6 miles below the surface of the Pacific Ocean, where pressure crushes military submarines flat. Something is alive. A single cell, the size of your fist. Feeding on uranium, scientists called those depths a dead zone. They were wrong. We are going to meet the creature that broke biology, find an entire city of animals that eats poisonous gas, and follow that discovery all the way to the frozen moons of Jupiter. If you enjoy the journey, a like and subscribe keeps this channel alive. Now strap in. We begin. For most of human history, scientists believed the deep ocean was empty. And honestly, that made sense. Go deep enough into any ocean, and the sun disappears. The water turns black. The temperature drops close to freezing. The pressure builds so fast that most metal objects crumple like aluminum cans on the way down. By the time you reach 6 miles below the surface, the math looked brutal. Pitch dark, near freezing. Pressure strong enough to crush a car into something the size of a shoebox. No food falling from above. No light to grow plants. Nothing a living creature could use. So for a long time, scientists drew a mental line, a depth limit. Below a certain point, they figured, life just stops. The ocean floor at extreme depths was labeled a biological desert. Scientists assumed it held nothing but sediment, rock, and maybe a few microscopic bacteria too simple to care about pressure or cold. That assumption lasted for decades. It filled textbooks. It shaped how marine biologists thought about where to look and where to stop looking. Then came the cameras. In the 1960s, a crew of engineers and scientists built a machine called the Trieste. It was a deep-sea vessel, round and heavy, designed to go where nothing had gone before. In January of 1960, 2 men climbed inside and descended into the Mariana Trench, the deepest known point on Earth. The trip took nearly 5 hours to reach the bottom. When they finally touched down, they expected emptiness. They expected rock and cold and nothing else. Instead, they saw a flatfish, a living animal, sitting on the ocean floor at nearly 7 miles down, calm as anything, like it had been waiting for company. That single fish cracked the old belief wide open. If a fish could survive down there, something was wrong with the theory. Scientists started asking harder questions. They started sending better cameras. They started looking instead of assuming. But here is where the story gets strange. The fish turned out to be the easy part. Over the following decades, as technology improved and cameras got sharper, scientists began finding creatures that did not follow any known biological rulebook. Animals with no eyes and no mouths. Organisms that ate toxic chemicals instead of food. And then, in 2011, a team from the Scripps Institution of Oceanography captured footage of something that stopped the entire field cold. A single cell, one cell, sitting on the floor of the Mariana Trench at 6.6 miles down. Bigger than your fist, bigger than most people's hands. A cell is supposed to be microscopic. You need a magnifying glass to see most of them. Every biology class teaches you that. A human body has trillions of cells, and the largest ones are still invisible to the naked eye. But this thing, this single organism made of a single cell, sat on volcanic rock 6 miles underwater, visible to a camera from several feet away. Scientists did not have a clean explanation. They still do not have a complete one. What they did know was this: the rule that said deep water meant no life was finished. A new set of rules was needed, and the deeper researchers looked, the weirder those new rules turned out to be. The creature had a name. It had a diet that should be impossible. That comes next. Stand in a swimming pool up to your waist. You can feel the water pressing against your legs. That pressure is mild, almost pleasant. Walk out to 10 feet deep, and your ears start to pop. Go down 30 feet, and your chest tightens. The pressure builds fast. Now picture going 6 miles deeper than that. At the surface of the ocean, the weight of the air above you pushes down at about 14 pounds per square inch. That is the baseline. Every living thing on land is built to handle exactly that amount of pressure without even noticing. Descend 100 feet into the ocean, and the pressure triples. Descend 1,000 feet, and it reaches roughly 450 pounds per square inch. At that point, an unprotected human body would be in serious danger. Blood vessels would struggle, lungs would compress. The math becomes violent fast. At 6 miles down, the pressure hits 8 tons per square inch. 8 tons, that is 16,000 pounds, pressing on every single square inch of anything sitting on that ocean floor. To picture 8 tons, imagine taking 50 full-grown adult elephants and stacking them on a surface the size of a postage stamp. Every square inch of the ocean floor down there carries that load. Every millimeter of a creature's body faces that force from every direction at once. A standard military submarine is built from thick reinforced steel. Engineers design it to handle enormous pressure. At 6 miles down, that submarine would be crushed instantly, folded like a paper cup in a fist. This is why the discovery of living creatures down there feels so wild. The pressure alone should be enough to destroy cells. Cells rely on membranes, thin, flexible walls that hold everything inside together. Extreme pressure deforms those membranes. It squeezes the water out of proteins and makes them useless. It shuts down the chemical reactions that keep cells alive. Yet the creatures living at 6 miles down are completely fine. Scientists have spent years trying to understand how. Part of the answer lies in a molecule called trimethylamine oxide, which deep-sea animals produce in large amounts. This molecule acts like a pressure stabilizer inside cells. It keeps proteins folded correctly. It prevents membranes from collapsing. The deeper the animal lives, the more of this molecule it carries in its body. Confirmed science. Multiple studies. The deeper you go, the more of this stabilizer you need, and deep-sea creatures produce it automatically. But trimethylamine oxide alone does not explain everything. Xenophyophores, the giant single-celled organisms found at 6.6 miles, do not seem to follow the same rules as other deep-sea creatures. Their cells are enormous, their structure is wildly different from anything found at shallower depths, and the pressure they survive every second of their lives would vaporize most living things. Researchers believe their structure plays a role. Xenophyophores build a rigid external frame from sediment particles they collect from the water around them. That frame may distribute pressure across the cell instead of letting it concentrate at any single point. A theory, a good one, but still a theory. What is confirmed is that these organisms sit at the most pressurized address on the planet, and they grow, feed, and live without any sign of struggle. The pressure that would destroy a submarine is, to them, simply Tuesday. And what they eat on that Tuesday is something else entirely. On January 23rd, 1960, 2 men sat inside a steel sphere barely big enough to turn around in and began sinking toward the deepest point on Earth. Their names were Don Walsh, a United States Navy lieutenant, and Jacques Piccard, a Swiss ocean engineer. The vessel they rode in was called the Trieste. It looked nothing like what most people picture when they imagine a submarine. The Trieste was a bathysphere cave, which means it floated using a massive tank of gasoline above the cruisphere instead of displacing water like a traditional sub. The gasoline was lighter than water, and it kept the whole machine from sinking too fast. The descent took 4 hours and 48 minutes. For most of that time, Walsh and Piccard sat in near total darkness, watching instruments and listening to the vessel creak around them. The Trieste groaned under the building pressure. At one point, a sharp crack echoed through the sphere. They had no way of knowing if the hull had split. They had no way of contacting the surface in real time. They just waited. The crack turned out to be the outer observation window cracking, a non-critical part of the structure. They kept going. At 6.8 miles down, the Trieste settled onto the floor of the Challenger Deep, the lowest known point in the Mariana Trench. Walsh and Piccard became the first humans in history to reach the deepest place on the planet. They had 20 minutes on the bottom. The instruments were limited. Visibility was almost zero because the landing stirred up sediment. But through the porthole, they observed a flatfish, resting on the floor as if the pressure and darkness were normal. That sighting, later questioned by some scientists who thought the animal might have been a sea cucumber or a different species, still confirmed one critical fact. Something was alive down there. The Trieste then began its slow ascent. The return trip took 3 hours and 15 minutes. Walsh and Piccard surfaced alive, their data intact, and the record they set that day stood for more than 50 years. No human returned to the Challenger Deep until 2012, when filmmaker and explorer James Cameron made a solo dive in a custom-built vessel. The 1960 dive changed ocean science permanently. Before the Trieste, the deep trench floor was largely a mystery. After it, scientists knew that reaching those depths was survivable, that life existed there, and that the ocean floor was worth studying. But the Trieste could only watch. It had no sample collection tools capable of pulling up the organisms from the floor. It could observe, record depth and pressure, and return. The real biological discoveries had to wait for better technology. Decades passed. Camera systems improved. Remotely operated vehicles got smaller and more precise. Scientists began sending machines instead of people, machines that could hover close to the floor, film in high detail, and collect samples without disturbing the surrounding area too much. When those machines finally reached the deepest zones with enough clarity to see what was actually living there, what they found required a complete rethink of cellular biology. A creature the size of a human fist, made of a single cell, was sitting on the floor waiting, and the way it built its body was stranger than anyone had guessed. Trace any food chain on Earth back far enough, and you hit sunlight. A lion eats a zebra. The zebra eats grass. The grass grows because of the sun. Swap the animals, change the ecosystem, it does not matter. Sunlight is the original energy source for almost every living thing on this planet. Remove it, and the chain collapses. This is why the deep ocean seemed so hopeless to early scientists. Sunlight starts losing strength the moment it hits water. At around 600 feet below the surface, almost no usable light remains. Scientists call the zone below this point the midnight zone. Total darkness. Permanent. Unbroken by seasons, storms, or any source of light, except the faint biological glow some deep-sea creatures produce themselves. Below the midnight zone, life thins out fast. Animals survive mostly by catching what falls from above. Dead plankton, pieces of dead fish, scraps of organic material drift down slowly from the sunlit upper ocean, like a very slow, very thin rain. Scientists call this marine snow. It is the only food source reaching the deep for most creatures. By the time marine snow drifts down to 4 miles, it has been picked apart by so many organisms along the way that almost nothing edible is left. A team studying deep-sea food webs calculated that only about 1 to 2% of the organic material produced near the surface ever reaches the deep trench floor. 1 to 2%. Enough to feed a few bacteria, hardly enough to support complex animals. So the scientific logic was clean. No light means no plants. No plants means no complex life. The deeper you go, the less there is to eat, and at 6 miles down, there should be almost nothing at all. That logic held for a long time. Then scientists found animals that completely ignored it. The creatures in the deepest trenches are not waiting for marine snow. Many of them are building their own energy from scratch, using chemical reactions instead of sunlight. But even before that discovery shook everything up, the discovery of xenophyrophores was already forcing a rethink, because a single cell that size cannot survive on trace scraps of drifting organic material. Xenophyrophores feed by a process called phagocytosis. They extend parts of their body outward and engulf particles from the surrounding water, pulling sediment, bacteria, and microscopic organic material directly inside themselves. They are slow, patient collectors. Their enormous size gives them a larger surface area to pull in food, which may be exactly why being big is an advantage in a place where food is so scarce. Confirmed science: larger surface area allows greater feeding range in low food environments. But the truly wild part of their diet is what else gets pulled in along with the food. Heavy metals, radioactive elements, toxic compounds that would wreck the biology of almost any other organism on the planet. Xenophyrophores do not avoid these materials. They accumulate them in extraordinary concentrations, and somehow they are completely fine. Understanding why starts with understanding what the inside of one of these creatures actually looks like, because it breaks every rule about what a single cell is supposed to be. In 2011, a team from the Scripps Institution of Oceanography was operating a remotely operated vehicle over the floor of the Serena Deep, a section of the Mariana Trench off the coast of Guam. The vehicle's cameras were scanning the sediment at 6.6 miles below the surface when the team spotted something unusual. Objects on the ocean floor, pale and irregular in shape, some reaching 4 inches or more across. They were resting on volcanic rock, motionless, scattered across the terrain like forgotten pieces of something. The team confirmed what they were looking at: xenophyrophores, giant single-celled organisms, at a depth that broke the previous record by more than half a mile. The record depth for finding xenophyrophores had just been shattered by a significant margin, and what the cameras were showing was something that biology textbooks were not built to explain. A cell is supposed to be tiny. Every biology class starts with that. The cell is the smallest unit of life. You cannot see most cells without a microscope. Red blood cells are so small that thousands fit across the width of a single hair. Even the largest human cells, like egg cells, measure less than a millimeter across. A xenophyrophore breaks that completely. A single xenophyrophore can grow to 4 inches in diameter. Some may grow larger. That makes them the largest individual cells ever documented on Earth. One cell, visible from across a room, the size of a tennis ball in some cases. This is the creature that was sitting on the floor of the deepest trench in the world, in total darkness, under 8 tons of pressure per square inch. The name xenophyrophore comes from Greek roots meaning bearer of foreign bodies, which describes the creature well. These organisms build a rigid outer structure from particles they collect from their environment. Grains of sediment, shell fragments, pieces of mineral. They cement these foreign materials together into a kind of scaffolding around their body, creating a structure called a test. This test serves multiple purposes. It provides physical support, which matters a great deal when your body is a single cell trying to hold itself together at enormous size. It may also help distribute pressure across the organism's surface, though scientists are still working out the exact mechanics. Inside the test, the cell itself is remarkable. Xenophyrophores contain thousands of nuclei inside a single cellular boundary. Most cells have one nucleus, the control center that holds the genetic material. Xenophyrophores have thousands of them, all sharing the same cytoplasm, the gel-like interior fluid of the cell. Biologists call this arrangement multinucleate. It is rare, and it raises a question that nobody has fully answered yet. Why does one cell need thousands of control centers? One theory is that the thousands of nuclei allow different sections of the cell to operate semi-independently, coordinating feeding, repair and reproduction across a structure too large for a single nucleus to manage. A theory, an interesting one. What scientists confirmed was that these organisms were alive, growing, and reproducing at the deepest point where any multicellular-looking creature had ever been found. They were also accumulating something inside their bodies that should have been killing them. And when the research team found out exactly what that was, the story got significantly stranger. Every cell in your body has a job, and part of that job is staying the right size. There is a reason cells are small. It is physics. As a cell grows larger, its volume increases much faster than its surface area. The surface is where the cell exchanges everything it needs with the outside world, pulling in oxygen and nutrients, pushing out waste. When the volume gets too large relative to the surface, the exchange system breaks down. Nutrients cannot reach the center fast enough. Waste builds up. The cell chokes itself. This is why almost every cell in almost every living thing on Earth stays microscopic. The size limit is a physical constraint, like trying to cool a football stadium with a single window unit air conditioner. Beyond a certain size, the system fails. Xenophyophores should not work. By standard cell biology, a cell 4 inches across would die from its own inefficiency long before it ever reached that size. And yet, there they sit, at 6.6 miles deep, very much alive. Scientists have worked out a few reasons why these organisms break the size rule. First, xenophyophores live in extremely cold water. Near freezing temperatures slow down all metabolic processes. The organism does not need nutrients delivered quickly because it is not burning through energy at a normal rate. A slow metabolism means a slow demand for resources, and a slow demand for resources means the surface-to-volume problem becomes less deadly. Second, the cell is not packed tightly. A xenophyophores' internal structure contains large amounts of open space. The cytoplasm threads through a framework rather than filling the cell solid. This increases the effective surface area inside the organism and allows chemical exchange to reach much deeper into the cell's interior. Third, and confirmed by multiple studies, the external test that xenophyophores build around themselves does more than provide support. It acts as an additional boundary layer, trapping particles and creating a kind of microenvironment around the cell. This may help regulate what enters and exits the organism, reducing the pressure on the cell membrane itself. The combination of all 3 factors, slow metabolism, open internal structure, and a built shell, creates a situation where a cell that should be physically impossible manages to function. But there is still a piece the scientists cannot explain. The pressure at 6.6 miles should be deforming the cell membrane continuously. Even with trimethylamine oxide helping to stabilize proteins, the sheer weight of the water column above pushing down on every surface of the organism is staggering. How the cell membrane holds its shape under that load without rupturing remains an open mystery. Scientists have hypotheses. None have been confirmed as the full answer. What they have confirmed is that the organism grows, survives, and multiplies successfully down there. The biology works. The exact reason why the biology works is still being argued over in research papers and conference rooms. And while scientists argue about membranes and pressure physics, the xenophyophore itself is busy doing something even more surprising. It is eating things that should be poisoning it to death, and it has been doing that for a very long time. Something strange happens to life when you remove all the light. In the sunlit ocean, small is often an advantage. Tiny creatures move fast, hide easily, and burn very little energy. Speed and stealth matter when predators can see you, but strip the light away completely, and the rules shift. In total darkness, size starts to become useful in ways that seem backward at first. Xenophyophores are the most extreme example, but they are not alone. Giant isopods, crustaceans related to the pill bugs you might find under a rock in your backyard, grow to roughly 16 inches long in the deep ocean. Their surface cousins measure about an inch. Deep-sea squid reach lengths that dwarf their shallower relatives. Giant sea spiders, animals with spindly legs and small bodies, reach leg spans of over 2 feet in the deep ocean, while the same family of creatures stays tiny near the surface. Scientists call this pattern deep-sea giantism, and it shows up across many different animal groups. The leading theories for why it happens are still debated, but several factors appear consistently across research. Food scarcity is one. When food is rare and unpredictable, a larger body can store more energy reserves. A bigger animal can go longer without eating and travel farther to find food when it is available. In an environment where a meal might not come for weeks, the ability to wait matters enormously. Slower metabolism is another factor. Cold water slows chemical reactions inside cells and tissues. A slower metabolism means growth continues over a much longer time period than it would at warmer temperatures. Animals in the deep can keep growing for decades longer than their surface relatives, which gives them time to reach sizes that would be impossible in warmer water. Pressure may also play a role. Some researchers think the physical compression of deep water pushes biological structures toward forms that are more compact, more efficient, or more stable under load. A larger, more robust body may simply hold up better than a fragile, smaller one. For xenophyrophores specifically, size appears to solve the feeding problem directly. A 4-inch cell has far more surface area to collect particles from the surrounding water than a microscopic cell would. In an environment where food drifts by slowly in extremely low concentrations, a confirmed observation, xenophyrophores in deeper, more extreme environments tend to be larger than those found at shallower depths. The relationship between depth, food scarcity, and body size appears to be real, though the precise biological mechanism driving it in xenophyrophores remains a subject of active research. And here is the piece that makes this genuinely unsettling. The particles that xenophyrophores collect from the surrounding water are not just organic scraps. The deep ocean sediment contains heavy metals that have settled out of the water column over millions of years. Sediment at trench depths carries measurable concentrations of lead, mercury, and uranium, elements that are highly toxic to most living things. A xenophyrophore, with its enormous surface area constantly sweeping particles from the surrounding water, is pulling in all of it, the toxic material alongside the nutritious material, with no apparent filter separating the two. What happens to all that poison inside the cell is one of the stranger things happening anywhere on this planet. Pull open any textbook on cellular biology and find the section on toxins. It will tell you correctly that heavy metals are some of the most destructive substances a cell can encounter. Lead attacks the nervous system and disrupts the enzymes that build proteins. Mercury binds to cellular proteins and warps their shape until they stop working. Uranium, a radioactive element, damages cellular structures directly through radiation, while also acting as a chemical toxin. Even trace amounts of these elements cause measurable harm to human cells. Scientists have documented exactly how much lead it takes to interfere with brain development, how much mercury accumulates in fish tissue before it becomes dangerous to eat. The thresholds are small, the damage is real. Xenophyrophores contain concentrations of lead, uranium, and mercury that dwarf anything scientists would consider safe for other living things. These are confirmed measurements from laboratory analysis of collected samples. The heavy metals are present at extraordinarily high levels inside the organism's test and cytoplasm, and the organism shows no signs of damage. Scientists currently classify this as a theory rather than a confirmed mechanism, because the internal biology that allows this to happen has not been fully mapped. But the evidence pointing toward high metal tolerance is strong and comes from multiple independent studies. One leading explanation focuses on how xenophyrophores store the toxic material. Many organisms that accumulate heavy metals, certain plants, some bacteria, do so by isolating the toxic elements inside specific compartments away from sensitive biological machinery. The metal gets locked up where it cannot interfere with proteins, enzymes, or genetic material. Xenophyrophores may be doing something similar on a massive scale. Their internal structure is complex and contains regions separated from the main cytoplasm by membranes. If toxic metals get directed into these isolated regions, they could accumulate in enormous quantities without reaching the parts of the cell that need to function. A second explanation involves proteins that specifically bind to heavy metals and neutralize their chemical activity. Some deep-sea organisms have been found to produce unusually high levels of metal-binding proteins. These proteins grab onto toxic metal ions before they can react with anything important. rendering them chemically inert inside the cell. Whether xenophyrophores use one of these mechanisms, both, or something entirely undiscovered, is still unknown. What is known is the result. These organisms act as natural sponges for some of the most toxic materials on the ocean floor. They pull heavy metals out of the sediment and water column and lock them inside their bodies. Researchers studying deep-sea contamination have noticed that xenophyrophores appear to reduce measurable heavy metal concentrations in the sediment around them. A creature that can clean up one of the most toxic environments on Earth by simply existing there is worth paying close attention to. But while scientists were puzzling over toxic-eating giant cells, a completely separate discovery was being made in a different trench. A discovery that did not just challenge one rule of biology, it challenged the entire assumption about where energy for life must come from. That story begins with a gas that smells like rotten eggs and a crack in the ocean floor. Picture a water filter, the kind people put on their kitchen taps. Water flows in carrying dissolved minerals, traces of chlorine, and microscopic particles. The filter catches the unwanted material and holds it while clean water passes through. The filter absorbs the problem so the system downstream stays clean. Xenophyrophores work something like that, except at a scale that covers the most extreme environment on the planet. The deep trench floor is not a clean place. Millions of years of geological activity have concentrated heavy metals in trench sediments. Hydrothermal venting at some locations pumps additional minerals and metals directly into the water column. Human industrial activity, while mostly a surface problem, has pushed measurable contamination down into even the deepest ocean layers. Scientists have found microplastics and industrial pollutants in Mariana Trench sediments. Into this environment, xenophyrophores extend their feeding surfaces and pull in everything around them. Because they accumulate rather than process and excrete heavy metals, whatever they absorb stays inside them. The surrounding sediment loses measurable concentrations of toxic elements wherever xenophyrophore populations are dense. A team analyzing sediment samples from areas with high xenophyrophore populations found lower heavy metal concentrations in the sediment directly beneath and around the organisms, compared to areas without them. The organisms were drawing the metals out of their environment and holding them. This has real potential. Bioremediation is the process of using living organisms to remove or neutralize toxic contamination from an environment. It works on land using certain plants and bacteria. Scientists already use sunflowers to pull radioactive cesium from contaminated soil. Bacteria eat petroleum spills. The concept is proven and in active use. Deep-sea contamination has always been a different problem because the depths are largely inaccessible and the conditions are too extreme for most organisms that clean up toxins near the surface. Xenophyrophores handle those conditions naturally. They already live at the most extreme addresses on the planet. They already accumulate heavy metals in high concentrations without suffering harm. If scientists could understand the exact mechanisms that allow them to do this, those mechanisms could potentially be replicated in other organisms engineered for cleanup work. Confirmed observation: xenophyrophores reduce local heavy metal concentrations in their immediate environment. The mechanism by which they tolerate those accumulated metals internally, still a theory, still being studied, but the practical implication is clear. A creature that can clean up radioactive and toxic contamination by sitting in it at pressures that destroy submarines is an organism worth understanding deeply. The question is whether the natural filter can survive long enough to keep working. Because the ecosystems these organisms live in are fragile in ways that took scientists a long time to appreciate. And in a different trench, several thousand miles away, scientists were finding a completely different kind of impossible. An entire world of creatures powered by poison gas, growing, feeding, reproducing, with zero input from the sun, zero input from the surface, and zero connection to anything happening 6 miles above them. Crack open a cell, any normal cell and you find one nucleus. That nucleus is the control center. It holds the entire genetic instruction set for the organism, the blueprint for every protein, every structure, every function the cell needs to run. One nucleus, one command center. That is the standard arrangement across almost all complex life on Earth. Xenophyrophores carry thousands of nuclei inside a single cellular boundary. This arrangement has a name: multinucleate. It exists in a small number of other organisms, some fungi, a few other giant amoebas, but nowhere else does it occur at the scale seen in xenophyrophores, and nowhere else does it occur under conditions this extreme. To picture what the inside of one looks like, imagine a building the size of a football stadium filled with a slowly flowing gel. Suspended inside that gel, spread across the entire volume, are thousands of small dense spheres. Each sphere is a nucleus. Each one carries a complete copy of the organism's genetic material. The gel flows slowly between them, carrying nutrients, signals, and cellular machinery from one area to another. Threading through this gel is a fine network of tubes and channels made from a material called plasmalemma, a type of specialized membrane. This network increases the internal surface area enormously, solving part of the problem of how to exchange nutrients and waste across such a huge volume. Between the nuclei, the cytoplasm also contains structures called mitochondria, the parts of the cell that convert food into usable energy. In a xenophyrophore, these are clustered in high concentrations near the outer feeding surfaces, where food particles are being pulled in and where the demand for energy to process them is highest. The outer shell, or test, that xenophyrophores build around themselves is not part of the cell itself. It is a construction made from foreign materials, cemented together by a substance the organism secretes. Scientists examining tests from deep trench specimens have found them built from tiny shell fragments, mineral grains, sediment particles, and occasional pieces of other organism structures. This test can be fragile or dense, depending on the materials available. In the Mariana Trench, where sediment composition differs from shallower areas, the tests of xenophyrophores found they have a distinct structure compared to specimens from other locations. The scientific classification of xenophyrophores has shifted over time. They were originally grouped with one category of organisms, then reclassified as a type of foraminifera, a group of single-celled organisms known for building protective shells. The current classification places them within the foraminifera, but acknowledges that their gigantism and multinucleate structure make them unlike anything else in the group. Every reclassification happened because the organism kept breaking the expected rules. Confirmed: thousands of nuclei, complex internal channel network, external test construction from environmental materials. Unknown: exactly how the thousands of nuclei coordinate their activity across the full volume of the cell. That coordination mystery is one scientists find genuinely puzzling. Communication within a single cell normally happens through chemical signals diffusing through the cytoplasm. In a 4-inch cell, diffusion alone is too slow to carry signals efficiently from one end to the other. Something else must be carrying information across the full volume. What that something is remains an open question. And while scientists puzzle over internal cell communication, the footage that confirmed these organisms existed at record depths created a moment that nobody in the research team expected. October 2011. A remotely operated vehicle descends into the Sirena Deep section of the Mariana Trench, operated by a team from the Scripps Institution of Oceanography. The vehicle carries high-definition cameras and a set of collection tools. The mission goal is broad survey work, documenting what organisms exist at extreme depths. The team had previous data from shallower surveys and some theoretical reasons to expect xenophyrophores might be present, but they were not specifically hunting for them at this depth. The vehicle reaches the floor at 6.6 miles down and begins its slow traverse across the sediment. Within the first pass, the cameras pick up unfamiliar shapes: pale, irregular, larger than anything the team expected to find at that depth. The vehicle's pilot moves in closer. The cameras resolve the objects clearly. Xenophyophores, dozens of them, spread across the volcanic rock floor in clusters, some sitting near the edges of small rock formations, others resting on flat sediment patches. The team's reaction, documented in the mission logs, was immediate recognition, followed by disbelief. Scientists on the research vessel watching the live feed began rapidly cross-checking depth readings against the current records for xenophyophore sightings. The previous record was roughly 6 miles. The vehicle was sitting at 6.6 miles. The new depth record was confirmed in real time. The lead researcher on the mission, Lisa Levin, described the discovery as one of those moments where the footage itself makes the argument before any analysis is done. The cameras showed large, complex, single-celled organisms at a depth that the scientific community had not expected to support them. The footage circulated through the marine biology community rapidly. The images were striking because of the sheer visibility of the organisms. These were creatures large enough to see clearly on camera from several feet away, sitting at the bottom of the deepest trench system on Earth. The paper published in the journal Biological Bulletin described the organisms in detail, confirmed the depth record, and raised several immediate questions. Were these the largest xenophyophores ever recorded? Were similar populations present in other deep trenches? How were they surviving at a pressure level that previous research had considered near the biological limit for complex single-celled organisms? The answer to the last question was, and remains, incomplete. What the footage established beyond argument was that the biological desert model of the deep trench floor was finished as a scientific consensus. Complex, large, actively living organisms were present at depths that should have stopped them. Every trench survey that followed found additional evidence supporting what the 2011 footage showed, and what those follow-up surveys began revealing was that xenophyophores were not alone down there. Something far more complex was growing on the floors of the deep trenches, an entire layered ecosystem running on an energy source that no biology textbook from the 20th century would have predicted. The cameras were about to show the science world something even harder to explain than a fist-sized single cell. Most people know the Mariana Trench by its deepest point, the Challenger Deep. That is the place that gets mentioned in comparison to Mount Everest, the place the Trieste visited in 1960. But the Serena Deep, a separate basin within the same trench system, is the section that has produced the most scientifically disorienting discoveries. It sits roughly 120 miles east of the Challenger Deep, and it reaches depths of 6.6 miles. The Mariana Trench itself runs about 1,500 miles long and 44 miles wide at its broadest point, roughly the length of the eastern United States from Maine to Florida, laid end to end. It curves through the western Pacific Ocean in a crescent shape formed by the collision of two tectonic plates. The Pacific Plate pushes beneath the Mariana Plate in a process called subduction, and the Mariana Trench is the crease in the ocean floor where that collision happens. Subduction creates something critical for the biology of the trench. As the Pacific Plate descends beneath the Mariana Plate, it carries seafloor sediment with it. That sediment is compressed under enormous pressure and heat, and some of the water, minerals and gases it contains get forced back up through cracks and vents in the overlying plate. This process brings chemical energy up from the Earth's interior into the trench environment. The Serena Deep sits over an area where this geological activity is particularly concentrated. The floor there is geologically active, with evidence of past and ongoing fluid movement through the rock. This creates a chemical landscape that is fundamentally different from a simple cold, dark, inert trench floor. Scientists sampling sediment from the Serena Deep have found elevated concentrations of specific compounds that indicate ongoing chemical reactions driven by deep geological processes. These compounds become food for organisms that can use chemical energy instead of sunlight. The combination of extreme depth, geological activity, and resulting chemical diversity makes the Serena Deep an environment unlike almost anywhere else on Earth. The pressure and darkness that define the experience of being there are joined by a constant slow chemical output from the rock itself. This is why xenophyrophores cluster there in high numbers. The geological activity produces the sediment conditions they need. The chemical richness of the floor supports the microbial populations they feed on alongside the mineral particles. And that chemical richness is also what supports a completely different class of organism, creatures that need no organic matter at all to survive, only the chemical energy locked in compounds seeping up from below. Confirmed, the Mariana Trench is formed by active subduction of the Pacific Plate. Confirmed, geological fluid activity in the Serena Deep creates elevated chemical concentrations on the floor. The full ecological map of which organisms are using which chemical sources and how they interact with each other remains largely incomplete. What has been mapped out is stunning enough, and what a team aboard a Chinese research vessel found in a different trench entirely just a few years ago suggested that the Serena Deep was only one chapter in a much larger story. 8 tons per square inch sounds like a large number. It is easier to understand when you break it into comparisons that actually land. 1 ton is 2,000 pounds. 16,000 pounds pushing down on a surface the size of a postage stamp. Now multiply that by every square inch of anything sitting on the ocean floor at 6.6 miles down. Take a submarine. A military submarine is built from high-strength steel roughly 2 to 3 inches thick in the hull, reinforced with internal structural frames, and designed specifically to resist water pressure. The average military submarine is rated to operate safely at a depth of roughly 1,500 feet. Deeper than that, and the hull starts deforming. At the depths of the Mariana Trench, a standard military submarine would be compressed into a small mass of crumpled metal in seconds. Styrofoam cups are a classic demonstration used on research vessel expeditions. Scientists attach styrofoam cups to the outside of deep-sea vehicles before descent. When the vehicle returns from the bottom, the cups have been compressed to roughly a quarter of their original size. The air pockets inside the foam collapse completely under the pressure. A cup that was 4 inches tall comes back 1 inch tall, dense and hard as plastic. The same physical force acts on every object at that depth, including the bodies of organisms living there. Living tissue is mostly water, and water does not compress the way air does. This is actually why organisms that live at depth are not immediately crushed. Water transmits pressure rather than collapsing under it, meaning the pressure inside the organism's body reaches equilibrium with the pressure outside. The organism is under pressure from all sides simultaneously, including from the inside. Places where tissues of different density meet, where gas might be trapped, where structures need to maintain a specific shape to function. Xenophyrophores have no gas-filled spaces. Their structure is liquid-filled throughout. This removes one of the main vulnerability points. But the cell membrane, the outer boundary of any cell, is still a flexible structure that has to maintain its integrity under 16,000 pounds per square inch. Acting on every point of its surface, the pressure is not coming from one direction. It comes from every direction at once, evenly, constantly. Scientists measuring the membrane properties of deep-sea organisms have found that their membranes contain higher concentrations of specific fats that remain fluid and flexible at high pressure. Surface organisms have membranes that stiffen and become rigid under the same conditions. The deep-sea versions are chemically adapted to stay functional. Confirmed, deep-sea organisms modify their membrane composition to maintain flexibility under pressure. Confirmed, trimethylamine oxide increases proportionally with depth across measured deep-sea species. Unknown, the full set of adaptations that allow xenophyrophores specifically to maintain structural integrity at 6.6 miles. The pressure at that depth is so extreme that it actively shapes which chemistry is possible. Some reactions that happen easily at the surface become impossible under high pressure. Some reactions that are rare at the surface become easier. The organisms down there have evolved to work within a different set of physical laws. And one group of organisms found slightly shallower, in a different trench, appears to have evolved an entirely different solution to the problem of survival. They simply stopped needing the surface at all. A xenophyrophore is one cell, but it is a cell that other creatures move into. The external test that xenophyrophores build around themselves, that rigid scaffolding of cemented sediment particles, creates a physical structure on the otherwise flat, featureless ocean floor. In an environment where hard structures are rare, any surface that rises from the sediment becomes valuable real estate. Scientists examining footage and collected samples from xenophyrophore-rich areas of the Mariana Trench have consistently found other organisms living on and inside the test structures. Small worms, tiny crustaceans, bacteria covering the inner surfaces of the test chambers, juvenile animals that appear to shelter inside the hollow spaces within the organism's external framework, confirmed observation. Multiple other species shelter within xenophyrophore test structures. The relationship appears to span several types. Some organisms are simply using the physical structure as shelter. The test provides a hard surface to attach to, a degree of protection from whatever currents move through the trench, and a three-dimensional space to hide within. These organisms are tenants in the most basic sense. They are there because the structure is there. Other organisms appear to be feeding on the xenophyrophore itself, or on the microbes living on its surfaces. The mucus-like material that holds the test together, and the bacterial films that colonize the outer surfaces of the test, represent a food source that small scavengers can access. Scientists have observed small worms in positions consistent with grazing on the test material. A third category involves organisms that appear to be using the xenophyophores' feeding activity to their own advantage. When a xenophyrophore sweeps particles from the surrounding water, it stirs the chemical environment slightly. Small organisms positioned near an active xenophyrophore may benefit from the disrupted sediment and the microbial activity that the xenophyophores' presence stimulates. The xenophyrophore, for its part, does not appear to actively defend its structure or drive away the tenants. Whether it benefits from any of these relationships is unclear. What the arrangement produces is a layered community of organisms in a location that would otherwise offer almost nothing to anchor to or feed on. The xenophyrophore creates a small oasis structure by simply being large and building a complex shell. This is the first hint at something that becomes much clearer when scientists look at the chemosynthetic ecosystems found in the deepest trenches. Life at extreme depth does not just survive as isolated individuals. It organizes. Creatures depend on each other, shelter within each other, feed on each other, and create conditions that other organisms then exploit. The individual organism is remarkable enough. The ecosystem it participates in is something else entirely. And in the Kuril-Kamchatka Trench, a trench system running along the floor of the northwestern Pacific, a team aboard a Chinese research submersible found an ecosystem so dense, so organized, and so completely self-contained that it rewrote what scientists believed was possible at extreme depth. They found it in the complete darkness of nearly 6 miles down, and it was enormous. Here is something worth holding in your mind for a second. Almost every calorie of energy in every living thing on Earth traces back to a photon of sunlight hitting a leaf. The sun hits the plant converts that light into sugar. Something eats the plant. Something else eats that thing. The chain runs through every ecosystem, every food web, every corner of the planet's surface. Remove the sun, and the entire system from grass to elephants to bacteria in your gut collapses. That was the assumption, a reasonable one, supported by every ecosystem scientists had ever studied in detail. Then scientists found hydrothermal vents in 1977, deep cracks in the ocean floor, where superheated water and chemicals pour out from the Earth's interior. Around these vents, thriving ecosystems existed in total darkness. no plants, no photosynthesis, no connection to the surface world at all. The organisms living there were pulling energy from chemicals, specifically from hydrogen sulfide, a toxic gas that smells like rotten eggs and would kill a human in high concentrations within minutes. The process is called chemosynthesis. Certain bacteria convert the chemical energy stored in hydrogen sulfide and methane into the sugar molecules that living things use for fuel. These bacteria are the base of the food chain. Other organisms eat the bacteria or host them inside their own bodies and feed off the products they generate. A food chain powered entirely by toxic gas and deep geological heat, running independently of the sun. Confirmed, chemosynthetic ecosystems exist around hydrothermal vents, confirmed by multiple expeditions since 1977. Those vent ecosystems were already remarkable, but the vent communities existed at depths of roughly 1 to 2 miles, not at the extreme trench depths of 5 to 6 miles. The trench floor is a different environment from a vent. Vents are geologically active, with concentrated chemical output from specific points in the rock. Trench floors are generally colder, quieter, and further from active geological hotspots. Scientists assumed chemosynthetic communities at the deepest trench depths were unlikely. The geological conditions seemed less favorable. The chemical output from the rock would be diffuse and scattered, not concentrated enough to support a rich ecosystem. In 2025, a team led by Mengran Du, a marine geochemist, proved that assumption wrong. Their submersible descended nearly 6 miles into the Kuril-Kamchatka Trench, a trench system running along the boundary between the Pacific Plate and the Okhotsk Plate in the northwestern Pacific. What the cameras found on the floor was a dense, sprawling community of organisms covering large sections of the rock surface. Tube worms, clams, microbial mats covering the rock like a white blizzard had settled on the ocean floor. All of it powered by chemicals seeping up from below, the deepest chemosynthetic ecosystem ever recorded by a significant margin. And the organisms living there had features that should have made survival impossible by any previous standard. What the tube worms looked like up close was the next thing the cameras revealed. The Fendouzay is a Chinese research submersible capable of reaching depths of nearly 7 miles. Its name translates to striver in English. In 2024, the Fendouzay descended into the Kuril-Kamchatka Trench carrying a science team that included Mengran Du, a marine geochemist from Shanghai Jiao Tong University. The mission goal was geological and chemical sampling. The team was primarily interested in the chemistry of the sediment and the fluids seeping up from the rock. The biology was secondary. The cameras changed the priorities. Within the first dive, the imaging system on the Fendouzay captured dense clusters of organisms covering sections of the volcanic rock floor, tube worms rising from the rock surface in groups, pale clams arranged across the sediment, white microbial material coating the rock like a layer of foam. The team had not expected to find a complex community at this depth. The Kuril-Kamchatka Trench sits between 5.4 and 6.3 miles deep across its main section. The area the Fendouzay was surveying was at the deeper end of that range, approaching 6 miles down. Multiple dives followed. Each one added more data. Scientists mapped the distribution of the community across the rock surface. The ecosystem was not a small, isolated patch. It covered a significant area of the trench floor, with different organism types occupying different zones within the habitat. The research team collected samples, recorded video, and took chemical measurements of the surrounding water and sediment. The chemical data showed elevated concentrations of methane and hydrogen sulfide seeping up through the rock in the survey area, consistent with geological fluid flow from the subducting plate below. The biology was using those chemicals as its primary energy source. The team's findings were published in the journal Nature in 2025. The paper described the community as the deepest high-biomass chemosynthetic ecosystem ever documented. Biomass is a measure of the total mass of living material in a given area. High biomass at this depth was the finding that drew the widest attention from the scientific community. Previous deep trench surveys had found scattered individual organisms, sparse bacteria, occasional animal traces. The expectation for the extreme trench floor was low density, slow activity. The Fendougé footage showed something that looked more like an underwater city than a biological desert. Dense populations, layered structure, different species occupying different niches within the same habitat. Confirmed, the deepest known high biomass chemosynthetic ecosystem exists in the Kuril-Kamchatka Trench. Confirmed, chemical analysis of the area shows methane and hydrogen sulfide seepage consistent with geological fluid flow. Unknown, the full spatial extent of the ecosystem, how long it has existed, and whether similar communities exist in other deep trench systems that have not yet been surveyed. The fact that this was found during a mission focused on geology, not biology, carries its own weight. Scientists were looking for chemistry. They found a city. The trench floor coverage by high-resolution cameras remains less than a fraction of 1% of the total deep trench area on Earth. Almost the entire deep trench environment has never been imaged. The creatures living in that city had bodies unlike anything found closer to the surface. A marine tube worm looks like something between a flower and a pipe. The body is a long, flexible tube anchored to a hard surface. At the top, a plume of filaments fans out into the water, red or white or pale depending on the species, swaying with any current. The overall impression, from a distance, is almost decorative, like something placed there by design. Look closer, and you find the biology that makes it genuinely strange. Marine tube worms have no eyes, no mouths, no digestive system of any kind, no stomach, no intestines, no pathway for taking in food and processing it in any way that resembles how animals normally feed. A human body, a fish body, an insect body, all of them share the same fundamental feeding design. Mouth takes in food, digestive system breaks it down. Nutrients absorbed into the bloodstream, waste expelled. Every animal on Earth follows some version of this plan. Tube worms abandoned it entirely. Inside a tube worm's body, taking up a large portion of its internal volume, is a specialized organ called a trophosome. The trophosome is packed with bacteria, specifically the chemosynthetic bacteria that convert hydrogen sulfide and methane into sugars. The tube worm does not eat the bacteria, it houses them. And the bacteria pay rent in the form of sugar molecules, the chemical fuel the tube worm cells need to function. The arrangement is called endosymbiosis, meaning symbiosis that happens inside the body. The bacteria live within the tube worm's tissues, protected from the external environment, and given access to the chemical supply the tube worm delivers to them. The tube worm lives off the sugars the bacteria produce. The tube worm's plume, the fan of filaments at the top of the tube, is the delivery system. It absorbs hydrogen sulfide and methane from the surrounding water and channels those compounds down through the body to the trophosome, where the bacteria use them. The plume also absorbs oxygen from the water, which the bacteria need to run the chemosynthetic reaction. Confirmed, deep-sea tube worms host chemosynthetic bacteria in a trophosome organ, confirmed across multiple species. The tube worms found in the Kuril-Kamchatka Trench community grow to roughly 12 inches long and cluster around areas where chemical seepage from the rock is highest. Their distribution maps almost perfectly onto the distribution of hydrogen sulfide and methane detected in the chemical surveys of the area. They find the gas, they anchor to the rock, they deploy their plumes, the bacteria inside them go to work. The clams in the same community operate by a closely related system. Their bodies also host chemosynthetic bacteria in their gill tissue rather than a trophosome, and the arrangement produces the same result. No need to find food, no need to hunt, no need for any connection to the surface world above them. These organisms measure up to 9 inches across, white-shelled, dense in the areas of highest chemical seepage. The ecosystem they are part of is self-contained in a way that no surface ecosystem can match. Sunlight goes out, and surface food chains collapse within weeks. This community would keep running. And what that means for the search for life on other worlds is something that changes the scale of the entire discussion. That comes next. The cameras on the Fendougé showed something that looked, at first glance, like snow. White material covered the rock surfaces across large sections of the survey area. It coated the stones in thick, irregular patches. It drifted in slow clouds near the seeps where chemical fluid pushed up from the rock. It settled over the sediment like a layer of frost on a winter morning. Scientists watching the footage recognized it immediately. Microbial mats. A microbial mat is a dense layer of bacteria and other microorganisms that builds up on a surface over time. In chemosynthetic environments, the bacteria that convert hydrogen sulfide and methane into sugar grow in such high concentrations around chemical seeps that their accumulated bodies form a visible coating on everything nearby. These are not individual cells floating loose in the water. These are colonies so dense that they become a physical layer, measurable in thickness, covering the rock the way moss covers a stone in a damp forest. The white color comes from sulfur compounds. Certain chemosynthetic bacteria produce elemental sulfur as a byproduct of their chemical reactions, and that sulfur accumulates inside and around their cells, giving the mats their pale, snowy appearance. A single square foot of active microbial mat can contain hundreds of millions of individual bacterial cells. The mat as a whole functions like a living carpet, constantly cycling chemicals from the seep through its biology and releasing different compounds back into the water. Confirmed, white microbial mats in chemosynthetic communities are formed by sulfur-oxidizing bacteria and related chemosynthetic microbes. These mats are the foundation of the entire ecosystem. Everything else in the community depends on them. The tube worms host chemosynthetic bacteria in their bodies, but those bacteria are closely related to the bacteria in the mats. The clams do the same. The mats represent the free-living version of the same biology, operating across the rock surface without a host. Smaller organisms graze on the mats directly. Amphipods, small crustaceans shaped roughly like shrimp, move across the mat surface, consuming bacterial cells. Polychaete worms thread through the mat material, extracting organic content. The mat supports a layer of grazers, who in turn support larger predators within the community. What the footage from the Kuril-Kamchatka Trench showed was this entire structure operating at nearly 6 miles down. The white material that looked like a blizzard was the base of a food web, dense, active, covering significant portions of the rock surface, producing energy from chemical seeps that will continue flowing for as long as the geological processes driving them remain active. Those geological processes are not dependent on anything happening at the surface. They run on the internal heat of the Earth, on the movement of tectonic plates, on chemical reactions happening miles below the ocean floor in the rock itself. The blizzard of microbes is the visible sign of an energy system that is completely independent of the sun, and when astronomers looked at that footage and then looked at their maps of the solar system, they saw something that shifted an entire field of research. Because 2 moons, one orbiting Jupiter and one orbiting Saturn, have conditions that match what the Fendougé cameras were filming. Almost exactly, in the Kuril-Kamchatka Trench at nearly 6 miles below the surface, the cameras found clams, white-shelled, dense, clustered around the edges of the microbial mats, and near the fissures in the rock, where chemical fluids seeped upward. Individual shells measured up to 9 inches across, roughly the diameter of a dinner plate. That size matters for context. Clams in shallow coastal waters typically grow to a few inches across over several years. Deep-sea clams in chemosynthetic communities grow more slowly because the energy available from chemosynthesis, while sufficient for survival, does not produce the excess that fast growth requires. A clam 9 inches across in the Kuril-Kamchatka Trench has likely been alive for a long time. Scientists studying clam populations at hydrothermal vent sites, which share their energy source with this trench community, have documented individual clams living for over 50 years. At trench depths, where temperatures are colder and metabolic rates slower, the lifespan of these organisms may extend significantly beyond that. A clam sitting at 6 miles deep in complete darkness with no connection to the surface world may be older than most adults listening to this. The shells themselves carry information. Growth rings in clamshells record periods of faster and slower growth, similar to the rings in a tree trunk. Scientists analyzing shells from chemosynthetic clam populations have used these rings to estimate ages and reconstruct the history of chemical seepage activity at specific sites. When the seepage was rich, the clams grew faster. When it slowed, the rings compressed. The shells in the Kuril-Kamchatka trench samples showed patterns consistent with long-term, stable chemical seepage from the rock. The community had not just appeared recently. It had been building for an extended period. The clam's feeding system mirrors the tubeworm's approach. Chemosynthetic bacteria live inside the clam's gill tissue. The clam circulates water across its gills, delivering hydrogen sulfide and oxygen to the bacteria. The bacteria produce sugars. The clam absorbs those sugars from the gill tissue directly into its bloodstream. No mouth involved in feeding at all. The mouth exists but is reduced and plays a minimal role. The gill system does the real nutritional work. Confirmed, deep-sea clams in chemosynthetic communities host bacteria in gill tissue and feed through endosymbiosis. Confirmed, clam shells from vent and seep communities show extended lifespans relative to shallow water species. Unknown, the exact age of the clam population in the Kuril-Kamchatka trench community. The discovery of large, old, healthy clams at nearly 6 miles down tells a specific story. This is a stable environment. The chemical seepage is consistent. The ecosystem has had time to mature. The organisms have had time to grow. A mature, stable ecosystem at 6 miles down, running entirely on geological energy, with no need for sunlight or surface input. The implications of that reach far beyond ocean science, because scientists studying the moons of other planets have been building an increasingly detailed picture of environments that look very much like this. Cold, dark, chemically active, sealed off from any external light source, and possibly full of life. The Kuril-Kamchatka Trench runs along the floor of the northwestern Pacific Ocean, stretching roughly 1,400 miles from the Kamchatka Peninsula down through the Kuril Islands toward Japan. It forms where the Pacific Plate slides beneath the Okhotsk Plate in a subduction zone, the same geological process that creates the Mariana Trench on the other side of the Pacific. As the Pacific Plate descends, it carries sediment and water with it into the deep rock. Pressure and heat squeeze the water and chemicals out of the descending plate, and those fluids push upward through cracks and fissures in the overlying rock. At the surface of the ocean floor, those fluids emerge as seeps, cold seeps, not the superheated vents found at mid-ocean ridges, but slow, steady releases of methane and hydrogen sulfide-rich fluid that percolate up through the sediment and rock. The survey area that the Fendougé team mapped was positioned over a section of the trench floor where this seepage was particularly active. Chemical measurements taken during the dives showed methane and hydrogen sulfide concentrations elevated well above background levels across the mapped area. The biological community tracked those chemical concentrations closely. Where seepage was highest, microbial mats were thickest. Tubeworms clustered densely around the most active fissures. Clams congregated at the edges of mat-covered zones. Further from the active seeps, the community thinned. The organisms followed the chemistry. Scientists mapped the community distribution against the chemical survey data and found a near-perfect correlation. Life clustered where the chemical energy was richest and thinned where it faded. This gradient structure is consistent with what scientists have found at hydrothermal vent communities and cold seep sites at shallower depths. The biology organizes itself around the energy source with remarkable precision. What made the Kuril-Kamchatka findings different was the scale and the depth. Previous cold seep communities had been found at depths of roughly 1 to 3 miles. A complex high-density community at nearly 6 miles down exceeded what existing models predicted should be possible. The pressure at that depth, the cold, and the assumed dilution of seep chemicals over the vertical distance from the geological source were all expected to limit biological complexity. The Fendougé data showed those limits were wrong. Confirmed, the biological community distribution in the survey area matched the chemical seep distribution measured during the expedition. The rock surface the community was built on is volcanic in origin, formed during past geological activity along the subduction zone boundary. The roughness and porosity of volcanic rock provides more surface area for microbial mats to colonize and more physical anchoring points for tubeworms and clams to attach to. Smooth sediment floor supports far less biological density in chemosynthetic communities. Rough volcanic rock dramatically increases the available habitat. The Fendougé team found their ecosystem on a section of the trench floor where the geology had produced exactly the right combination of chemical seepage and physical surface structure. The ecosystem that built up on that rock has been running, in total darkness, completely isolated from the surface world, for longer than any current estimate can pin down. And what runs underground can run under ice too. The astronomers had been watching closely. Pull a thread on any surface ecosystem, and it eventually leads back to the sun. A forest fire destroys a section of trees. Within years, grass grows back, birds return, deer follow. The recovery is driven by sunlight falling on whatever plants begin regrowing first. Remove the sun for long enough, and the recovery stops. A drought hits a grassland, rivers shrink, animals move or die. When rain returns, the grass comes back because the sun is still there to drive the growth. The resilience of surface ecosystems depends entirely on sunlight remaining available. The ecosystem in the Kuril-Kamchatka Trench has no such dependence. Its energy source is geological. The seepage of methane and hydrogen sulfide from the descending Pacific Plate is driven by tectonic forces that have been running for tens of millions of years and will continue running for tens of millions more. The movement of the Pacific Plate under the Okhotsk Plate is not going to stop. The chemical output from that geological process is not going to stop. The sun could go dark tomorrow, and the ecosystem in the Kuril-Kamchatka Trench would be completely unaffected. Nothing about the food chain there requires light, oxygen from the atmosphere, surface water, seasonal cycles, or any input whatsoever from the world above the ocean surface. Scientists call this type of ecosystem a chemoautotrophic system, which means it is self-sustaining through chemical energy from the Earth's interior. The prefix auto in that term is important. It means self. The system feeds itself. Confirmed, the energy source for the Kuril-Kamchatka Trench community is geological fluid seepage, independent of photosynthesis or surface inputs. The implications of a fully independent ecosystem extend into every field of science that asks questions about where life can exist. For most of human history, the assumed requirements for life were clean and simple: liquid water, suitable temperatures, energy from sunlight, organic chemistry. Find a planet or moon with those 4 things, and life becomes plausible. Miss any one of them, and life becomes unlikely. The deep trench ecosystem removes sunlight from that list entirely. Liquid water, yes. Suitable temperatures relative to the organism's adaptations, yes. Chemical energy from geological sources, yes. Organic chemistry built from those chemicals, yes. Sunlight, 0. This rewrite of the minimum requirements for life has direct consequences for how scientists evaluate other worlds in the solar system. Several bodies in the outer solar system carry liquid water oceans beneath thick crusts of ice. No sunlight reaches those oceans. No surface atmosphere interacts with them. By the old requirements, they were considered unlikely candidates for life. By the new requirements, they are candidates. The chemistry-powered ecosystem sitting in the dark at 6 miles down provided the scientific argument that changed the evaluation. The strongest candidate for extraterrestrial life in the solar system is not on a planet with blue skies and warm temperatures. It is under ice, and the case for it is built almost entirely on what was found at the bottom of the Pacific. Uranium is radioactive. Every nucleus of uranium that breaks apart sends out radiation, particles and energy that damage biological molecules on contact. Radiation breaks the chemical bonds in DNA. It knocks electrons out of proteins and warps their structure. It punches through cell membranes and disrupts the chemistry on both sides. In high enough doses, radiation destroys cells outright. In lower doses, it damages them in ways that accumulate over time, leading to mutations, cancer, and cellular failure. This is why exposure to high levels of radiation is life-threatening for humans. The biological damage is direct and measurable. Xenophyrophores accumulate uranium inside their bodies in concentrations many times higher than the surrounding sediment. This is a confirmed measurement. The radioactive element is present, it is concentrated, and it is inside the living cell. The theory is that xenophyrophores are highly resistant to radiation damage, possibly more resistant than any other complex organism known. If that theory holds up, it matters enormously. Scientists studying radiation resistance in biology have found a hierarchy of tolerance across different life forms. Humans tolerate a modest dose before cellular damage becomes critical. Many insects tolerate significantly higher doses. Certain bacteria, most famously a species called Deinococcus radiodurans, tolerate doses thousands of times higher than a human lethal dose and simply repair the DNA damage as fast as it occurs. Deinococcus radiodurans does this through an exceptionally rapid and accurate DNA repair system. Its machinery for fixing broken DNA strands is dramatically more efficient than anything found in human cells. If xenophyrophores have developed a comparable system to handle the uranium they accumulate, understanding that system could have direct applications. Cancer treatment, where the goal is often to use radiation to destroy tumor cells while limiting damage to healthy tissue, could benefit from understanding how to make cells more resistant to radiation damage. Long-duration space travel exposes astronauts to cosmic radiation at levels that current shielding cannot fully block. Organisms that handle radiation naturally, at the biological level, offer a starting point for research into solutions. A theory currently. The evidence supporting it is strong. The confirmed mechanism has not been worked out. Scientists have not yet fully mapped the internal biology of xenophyrophores at the resolution needed to identify a specific radiation repair system. The organisms are difficult to collect intact and nearly impossible to keep alive in laboratory conditions, which means most analysis happens on dead or fragmentary specimens. Unknown. The specific biological mechanism allowing xenophyrophores to tolerate accumulated uranium without detectable cellular damage. The gap between the observed fact and the understood mechanism is one of the most significant open questions in deep-sea biology. Filling that gap requires collecting live specimens and studying their active cellular chemistry. Collecting live specimens from 6 miles down, intact and functional, is at the current edge of what deep-sea technology can do. The answer may be sitting on the floor of the Mariana Trench right now. And while scientists work toward the tools to retrieve it, a completely separate puzzle about these organisms has remained unsolved for much longer. The frustration of studying xenophyrophores is that the most important questions are also the hardest to answer. Every confirmed fact about these organisms raises a new unknown. The cell is enormous. Why? Confirmed partial answers, but the full picture is missing. The cell tolerates crushing pressure. How? Partial mechanisms identified. Gaps remain. The cell accumulates uranium, lead, and mercury without damage. By what process? unknown. That last question is the one that has generated the most unresolved debate in the scientific literature. Heavy metal tolerance in biology is not completely new. Certain plants, called hyperaccumulators, grow naturally in soils with high heavy metal concentrations and pull those metals into their tissues in high amounts. Alpine pennycress is one example. It grows in zinc and cadmium contaminated soils and accumulates those metals in its leaves at concentrations that would be toxic to other plants. Scientists have spent decades studying how hyperaccumulators work. The mechanisms they have found involve specific protein pumps that move metal ions across cell membranes in controlled directions, storage proteins that bind metals and neutralize their chemical activity inside the cell, and compartment structures that physically separate accumulated metals from sensitive cellular machinery. These mechanisms work. They are confirmed and well understood in plants. Whether xenophyrophores use any of the same mechanisms is unknown. The two organisms are about as biologically different as it is possible to be. One is a plant with many cells. The other is a single-celled organism, more closely related to certain ocean-dwelling microorganisms than to any plant. The similarity in heavy metal accumulation behavior does not mean the mechanisms underneath are the same. Researchers have attempted to analyze the chemical forms of heavy metals inside xenophyrophore specimens. The goal is to determine whether the metals are present as free ions, which would be highly reactive and damaging, or as bound complexes with proteins or other molecules, which would indicate the organism has neutralized their chemical activity. Results from these analyses have been mixed. Some studies suggest the metals are present in bound forms, consistent with an active detoxification process. Others find patterns that are harder to interpret. The core problem is specimen quality. Xenophyrophores from the deep trench are fragile. The pressure difference between the ocean floor and the surface is so extremely that bringing specimens up intact is extraordinarily difficult. By the time a sample reaches the research vessel, cellular structure has often been compromised. The chemistry inside the cell has changed under the altered pressure and temperature conditions during ascent. Scientists are essentially trying to analyze a biological system that partially falls apart the moment they try to retrieve it. Confirmed, xenophyrophores accumulate heavy metals at concentrations far above background levels. Unknown, whether the accumulation represents active detoxification, passive absorption, or some novel mechanism with no equivalent in known biology. The answer is sitting at the bottom of the ocean, chemically intact, doing whatever it does, completely inaccessible to the tools currently available. New submersibles with pressurized sample containers designed to bring deep-sea specimens up without a pressure change are being developed. When they work reliably, this question may finally get an answer. Until then, the creature keeps its secret. And deeper in the trench system, the unknown piles up even higher. The surface of Mars has been mapped more thoroughly than the floor of Earth's oceans. That is a confirmed fact. Space agencies have produced detailed topographic maps of Mars covering the entire planet. The ocean floor of Earth, which covers roughly 70% of the planet's surface, remains less than 25% mapped at high resolution. The reason is physical. Mapping from space using radar and optical instruments works well for land and other planets. The ocean blocks those signals completely. Mapping the ocean floor requires sending sound waves down from ships, waiting for the echo to return, and building an image from the timing. This process is slow, requires ships to travel directly over every area being mapped, and at its best resolution, still produces images less detailed than what satellites deliver for land surfaces. The deep trenches are among the least mapped regions of all. Most trench surveys cover small target areas rather than comprehensive stretches of the trench floor. The Fendouje mission surveyed a section of the Kuril-Kamchatka Trench, found a dense biological community, and did not have the time or resources to determine how far that community extended beyond the surveyed area. Scientists currently estimate that less than 1% of the deep trench floor has been imaged at a resolution capable of detecting and identifying organisms the size of tube worms or clams. Less than 1%. The ecosystem found in the Kuril-Kamchatka Trench was found almost accidentally. During a mission focused on geology, the Serena deep xenophyophores were found during a broad survey not specifically targeting those organisms. The history of deep trench discoveries is a history of finding things no one predicted while looking for something else. The total number of chemosynthetic communities in the world's deep trenches is entirely unknown. The total biomass of xenophyophores across all trench systems has never been measured. The full range of species living in deep trench environments is a fraction of what actually exists. Confirmed, less than 1% of the deep trench floor has been surveyed at biological detection resolution. Unknown, the number, extent, and diversity of biological communities in unsurveyed deep trench areas. The trenches run across the Pacific floor in long arcs. The Mariana Trench, the Kuril-Kamchatka Trench, the Philippine Trench, the Tonga Trench, the Puerto Rico Trench in the Atlantic. Each of these systems is thousands of miles long. Each sits at depths between 4 and 7 miles. Each has been surveyed in only a handful of locations. What lives in the unsurveyed sections is genuinely unknown. And here is what makes that gap in knowledge significant beyond just ocean science. The argument for life on other worlds rests partly on the Earth analogy. If life exists in the most extreme environments on Earth, the argument goes, extreme environments elsewhere may also harbor life. The strength of that argument depends on how many extreme Earth environments we have actually checked. The answer for deep ocean trenches is almost none of them. We found a city of organisms at 6 miles down while looking for chemistry. The rest of the floor remains dark and unexamined. The next discovery could be sitting in a trench we have never pointed a camera at. 2 moons in the outer solar system have been sitting at the center of one of the biggest arguments in astronomy for the last 2 decades. Europa orbits Jupiter, Enceladus orbits Saturn. Both of them are covered in thick crusts of ice. Both of them have liquid water oceans underneath that ice, confirmed by gravitational measurements, magnetic field readings, and in Enceladus's case, active geysers of water vapor erupting from cracks in the surface and shooting into space. These are not theoretical oceans. They are real, confirmed, and large. Europa's ocean is estimated to hold roughly twice the volume of all Earth's oceans combined, sitting beneath an ice shell between 10 and 30 miles thick. Enceladus has a smaller ocean, but one that is demonstrably active. The geysers it sends into space carry water, organic compounds, silica particles, and hydrogen gas, all materials that indicate ongoing chemical reactions between the ocean water and the rocky seafloor beneath it. That seafloor detail is critical. Enceladus's ocean sits above a rocky core. The gravitational forces from Saturn stretch and squeeze the moon continuously, generating heat through friction in its interior. That heat drives hydrothermal activity on the seafloor, similar to the vent systems on Earth, but in a moon 400 million miles from the Sun. Hydrogen gas detected in the Enceladus geysers is a strong indicator of ongoing water-rock reactions at elevated temperatures, a process that generates exactly the chemical energy that Earth's chemosynthetic bacteria use. Confirmed, liquid water oceans exist beneath the ice of Europa and Enceladus. Confirmed, Enceladus geysers contain hydrogen, organic compounds and silica, consistent with seafloor hydrothermal activity. For most of the 20th century, the dominant view in astrobiology was that life requires proximity to a star. Planets in the habitable zone, the Goldilocks distance where liquid water can exist on a surface, were the targets. Cold outer solar system moons were ruled out. No sunlight, no surface liquid water, no life. The deep sea discoveries did not just nudge that view. They dismantled it. If an ecosystem can run at 6 miles depth on Earth, powered entirely by geological chemistry, with zero input from the sun, then the same could operate in the seafloor of Enceladus's ocean. The conditions match with uncomfortable precision. Cold liquid water sitting above chemically active rock. No sunlight. potentially hydrogen sulfide and methane available as energy sources. The deep trench communities of the Pacific Ocean are now used as the primary biological analog when scientists model what life in Enceladus might look like. The argument is not that life definitely exists there. The argument is that conditions known to support life on Earth exist there right now, and one mission, already in planning at the time of the Fendu's discovery, is being designed specifically to find out Europa is roughly the same size as Earth's moon. From a distance it looks smooth and bright white, covered in a clean sheet of ice, crisscrossed by reddish-brown lines. Those lines are cracks where the ice has shifted and refrozen repeatedly. They trace the surface like the cracks in a hard-boiled egg. Beneath the ice lies an ocean estimated to be between 40 and 100 miles deep. For comparison, the average depth of Earth's oceans is about 2 miles. Europa's ocean, if the deeper estimates are correct, is roughly 50 times as deep as Earth's average ocean depth, and it sits under an ice shell so thick that no sunlight has ever touched the water below. The conditions inside Europa's ocean, as far as scientists can determine from remote measurements, include liquid water, likely cold and under high pressure toward the seafloor, and a rocky floor below the water. The interaction between rocky seafloor and liquid water is where the chemistry happens. On Earth, ocean water reacting with rock at elevated temperatures produces hydrogen and methane through a process called serpentinization. The rock minerals, particularly olivine, react with the water and change their chemical structure, releasing hydrogen gas in the process. Chemosynthetic bacteria on Earth use that hydrogen as an energy source. Scientists analyzing Europa's likely rock composition and interior temperature models have calculated that serpentinization could be occurring on Europa's seafloor. The ocean water would react with the rocky floor in exactly the same way it does at Earth's mid-ocean spreading centers. A confirmed parallel, serpentinization is a known process on Earth that produces hydrogen used by chemosynthetic organisms. Europa's conditions are consistent with serpentinization occurring, though it has not been confirmed directly. The evidence from Enceladus is stronger because those geysers give direct access to material from that moon's ocean. Europa does not have accessible geysers, so the evidence for its ocean chemistry is more indirect. The Europa Clipper mission, launched by NASA in October 2024, is currently traveling toward Jupiter and is scheduled to arrive in 2030. It carries instruments designed to analyze Europa's magnetic field, ice surface chemistry, and gravitational profile in detail. The mission goal is partly to characterize the ocean beneath the ice and assess its habitability. Confirmed, Europa Clipper launched in October 2024 and is en route to Jupiter. The data it collects will either strengthen or weaken the argument that Europa's ocean could support life, but the argument itself was built on what scientists found in Earth's deep trenches. Without the xenophyrophores in the Mariana Trench and the tubeworm city in the Kuril-Kamchatka Trench, the case for Europa as a life candidate would be significantly thinner. The creatures at the bottom of the Pacific Ocean are, indirectly, the reason a spacecraft is right now traveling 450 million miles toward Jupiter. And the argument they built for the possibility of alien life is the strongest scientific argument for extraterrestrial biology ever assembled. Before the deep-sea discoveries, the search for extraterrestrial life focused almost entirely on surfaces. Mars had a thin atmosphere and evidence of ancient rivers. Venus was too hot. The outer solar system planets were gas giants with no solid surface. The search stayed close to the Sun, aimed at places where sunlight could reach the ground, and warm liquid water could sit in the open. Then the deep sea upended the surface bias. Life does not need warmth from a star. Life does not need oxygen or photosynthesis or any of the atmospheric conditions that make Earth's surface comfortable for humans. Those are requirements for the type of life that evolved on Earth's sunlit surface. They are not requirements for life itself. The scientific argument runs like this. Chemistry produces energy. Geology drives chemistry. Wherever geology is active, chemical energy is available. Wherever chemical energy is available in liquid water, life on Earth has found a way to use it. The xenophyrophores in the Mariana Trench use chemical energy indirectly, feeding on organisms and materials produced by geological processes. The tubeworm and clam community in the Kuril-Kamchatka Trench uses chemical energy directly, hosting bacteria that run the conversion themselves. These communities are not edge cases. They are not rare anomalies hiding at one unusual location. Multiple independent expeditions have found chemosynthetic communities at extreme depths in different trench systems across the Pacific. The pattern is consistent. Life found geology, life used the chemistry, life built stable, multi-generational ecosystems. The outer solar system has geology. Europa has a rocky seafloor with liquid water above it. Enceladus has a rocky seafloor, liquid water, and active hydrothermal output, already confirmed by its geysers. Titan, Saturn's largest moon, has liquid on its surface, though it is liquid methane rather than water. The confirmed existence of stable, complex ecosystems, powered entirely by geological chemistry on Earth, means that any body in the solar system with liquid water above active rock is a legitimate candidate for life. That list is longer than it was 30 years ago. Confirmed, chemosynthetic ecosystems at extreme depth have been found in multiple trench systems. Confirmed, Europa and Enceladus have confirmed liquid water oceans with evidence of rocky seafloor chemistry. The gap between confirmed Earth biology and confirmed extraterrestrial life remains total. No alien organism has been found. No extraterrestrial biological signal has been confirmed. But the argument that life might exist in Europa's or Enceladus's ocean is built on real evidence from real organisms in real ecosystems on Earth. That is different from theoretical speculation. Scientists studying astrobiology now routinely use deep trench biology as their reference point when evaluating candidates for life in the outer solar system. The creature at 6 miles down changed the conversation. What finding actual life on another world would mean for humanity is a question that goes far beyond science. Consider the number 1. If life exists only on Earth, the number of places in the universe where life has independently originated is 1. Earth. 1 example, 1 data point. With a single example, you cannot tell whether life is common, rare, or a freak accident that happened once in a trillion circumstances and will never happen again. 1 example tells you only that it happened once. Nothing about the probability. Find a second example anywhere, even microbial life in Europa's ocean, and the number becomes 2. 2 independent origins of life changes the math entirely. If life originated independently in 2 separate locations in the same solar system, the probability that life is a common outcome of chemistry in the universe stops looking tiny. If 2 places in one solar system developed life, and there are hundreds of billions of star systems in this galaxy alone, the reasonable conclusion is that life is not an accident. Life is something the universe does when the conditions are right. That conclusion would be the most significant discovery in the history of science. It would also raise the probability that intelligent life has arisen elsewhere, though the gap between microbial life and intelligent civilization is enormous and carries its own set of unknowns. The philosophical and religious weight of confirming life beyond Earth is significant enough that agencies studying astrobiology have dedicated resources specifically to the question of how to announce such a discovery responsibly, if it occurs. Confirmed, NASA and international space agencies have protocols under development for handling confirmed detection of extraterrestrial life. The scientific consequences would be immediate and far-reaching. Every model of how life originates would need to be checked against a second example. Every assumption built on Earth's single data point would become provisional. The entire field of biology would gain a comparative framework it has never had. Medicine, evolutionary science, ecology, biochemistry, all of them are built on the study of life on Earth. A second origin of life, using potentially different chemistry, different molecular structures, different cellular architecture, would either confirm that life's core mechanisms are universal, or reveal that multiple completely different solutions to the problem of staying alive exist. Either outcome would be extraordinary. The organisms in the deep Pacific trenches did not prove that life exists in Europa's ocean. They proved that the conditions in Europa's ocean are consistent with supporting life. The distinction is important, but they built the argument. They changed the question from, could anything live there, to what would we actually find if we looked? And the shift was driven by a creature the size of a fist, sitting in total darkness at the bottom of the world, eating uranium. One of the most argued questions about the Kural-Kamchatka Trench community is also one of the most basic. Where did the organisms come from? The tubeworm species found in the deep trench community belong to groups that are also found at hydrothermal vent sites at depths of 1 to 2 miles. The chemistry they use is the same. The body plan is the same. The endosymbiotic bacteria they host are closely related. So the question is whether the deep trench population descended from ancestors that were already living at shallower vent sites and gradually moved deeper over evolutionary time, or whether they arrived at the trench floor through some other pathway and adapted to conditions there independently. Migration seems like the simpler answer. Tubeworm larvae, the early life stage of the organism, are free floating in the water column for a period before they settle and anchor to a surface. A larva released near a shallower vent system could theoretically drift in deep ocean currents and eventually settle at a trench floor, if it encountered the right chemical conditions. The problem is distance and pressure. The larvae of known tubeworm species from shallow vent systems are adapted to the pressure and temperature of those shallower depths. A larva drifting from 1 mile down to 6 miles down would experience a pressure increase of roughly 30 times its starting conditions during that descent. Whether the larva could survive such a descent intact and functional is genuinely uncertain. No study has yet tracked the larval drift pathways for tubeworm species in the Pacific in enough detail to determine whether gene flow between the shallow vent populations and the deep trench population is occurring. An alternative pathway is colonization from organism pieces, or reproductive material carried down by the falling organic material, the marine snow, that drifts from shallower depths. Chemosynthetic bacteria are widely distributed in ocean sediments. If bacterial populations reach the trench floor through settling, and if chemical seeps emerge in the right locations, tubeworm larvae that arrive independently could find established microbial communities already present and ready to colonize. A third possibility is that the deep trench population has been isolated long enough to have evolved independently from the shallower species. Independent evolution under such different pressure conditions could produce organisms that look similar on the outside, but have different internal biology. Confirmed, the tubeworm species in the Kural-Kamchatka Trench community are closely related to species at shallower vent systems. Unknown. Whether this represents descent from a shared ancestor with gradual depth adaptation or independent colonization from deeper arrival pathways. The answer requires genetic analysis comparing the deep trench population to shallow vent populations across the Pacific. That analysis is ongoing. What the question reveals is that the deep sea is not a static dead zone. It is a dynamic environment where populations move, adapt, and potentially speciate over geological time. The trench floor is not just where things end up, it may be where entirely new things begin. And the timeline of these ecosystems, how long they have been running and how they have changed, raises a question that may be even harder to answer. Every biology textbook carries a list of requirements for life: liquid water, a source of energy, carbon-based organic chemistry, a set of stable chemical conditions that allow complex molecules to form and maintain their structure over time. These requirements were built from studying life on Earth. Every organism ever documented fits within them. The list felt solid. a checklist for finding life elsewhere, the deep-sea discoveries did not remove any item from the list. Liquid water, still required. Organic chemistry, still required, but they forced a rethink of the acceptable ranges for each condition. Water at 6 miles down is near freezing and under crushing pressure. The acceptable temperature range for liquid water capable of supporting life just expanded significantly below the comfortable range assumed by surface biology. Energy from geological chemistry rather than sunlight expanded the acceptable energy sources from 1 to at least 2, and possibly more. Scientists studying other forms of chemical energy available in geological environments, including iron ammonia and various mineral reactions, are now testing each as a possible life energy source. Pressure tolerance, demonstrated by xenophyrophores and deep trench animals, extended the physical range of conditions compatible with life far beyond what the surface records suggested. Toxic tolerance, demonstrated by organisms in chemosynthetic communities, expanded what counts as a chemically hostile environment. Organisms that not only survive but thrive in high hydrogen sulfide concentrations, high heavy metal concentrations, and near zero oxygen conditions challenged the idea that toxicity itself sets a hard limit on life. Each of these expansions has direct implications for the search for life elsewhere. Confirmed, deep trench organisms function at pressures, temperatures, and chemical conditions outside the ranges assumed from surface biology alone. The astrobiological impact is significant. When scientists model potential habitable environments on other worlds, they use Earth biology as the reference for what conditions life can handle. Every time deep-sea research expands that reference, the number of worlds that count as habitable candidates grows. Europa's cold, pressurized ocean fits within the expanded range. Enceladus's chemically active seafloor fits within the expanded range. Even more distant and unusual environments become plausible. The deep-sea creatures are not just interesting biology. They are the measuring stick that determines how broadly scientists are allowed to look. But this expansion of acceptable conditions also raises an uncomfortable question that scientists have been debating since the first chemosynthetic vents were found. If life is so broadly capable of finding energy wherever it exists, why is the deep trench environment so poorly explored? And if we have already found 2 major biological surprises there in just the last 15 years, what else is sitting in the dark, completely undiscovered, in the 99% of trench floor that has never been imaged? That question does not have a comfortable answer. The Challenger Deep is the deepest confirmed point on Earth, sitting at approximately 6.8 miles below the sea surface. The Fendougé ecosystem was found at nearly 6 miles. The xenophyrophore record sits at 6.6 miles. The Challenger Deep is deeper than both. The question of whether biological communities exist at the absolute deepest points of the ocean, right at the bottom of the Challenger Deep and comparable extreme points in other trenches, has not been answered. Sparse surveys of the Challenger Deep have found bacteria and individual organisms at extreme depths, but no high-density community comparable to what the Fendougé survey documented in the Kuril-Kamchatka Trench has been confirmed at the very deepest points. The absence of a finding is different from confirmation that nothing is there. The challenge at the absolute deepest points is that the geological conditions that support chemosynthetic communities, active seeping of methane and hydrogen sulfide from the rock, may not be present everywhere. The distribution of chemosynthetic communities depends on the distribution of geological fluid seepage, which is not uniform across the trench floor. Areas of the Challenger Deep that happen to sit over active geological seep zones could theoretically support communities similar to what the Fendougé found. Areas that lack active seepage would not. Confirmed, the Challenger Deep has been visited by human-occupied and remotely operated vehicles on a small number of occasions. Confirmed, bacteria and individual organisms have been documented at the deepest measured points. Unknown whether organized, high-density biological communities exist at or near the absolute deepest points of any trench system. The technology required to survey the entire Challenger deep floor at the resolution needed to detect tubeworm-scale organisms does not yet exist in deployable form. The vehicle capable of carrying adequate cameras and sample collection tools to the exact depth needed, hovering slowly across the floor with enough battery life to cover meaningful area, is at the frontier of what current ocean engineering can build. Several nations, including China, the United States, and Japan, have deep-sea programs actively developing more capable vehicles. The Fendougé itself represents one of the most advanced systems currently operational. The timeline for comprehensive deep trench surveys is measured in decades. In those decades, the pace of discovery is likely to continue, matching what the last 15 years have produced. Each mission finds something no one fully predicted. The pattern is consistent enough that scientists now plan deep trench expeditions with the expectation that they will find biology they did not expect, even if they cannot specify what it will be. The floor of the deepest places on Earth is not empty. That has been established. How full it is remains the open question. And the technology gap standing between current science and a real answer is the same gap that stands between us and the ocean floor of Europa. The engineering challenge of reaching the bottom of a trench on Earth and the engineering challenge of reaching the ocean floor of an ice-covered moon are different in scale, but similar in kind. The tools being built for one will help build the tools for the other. The Fendougé can reach nearly 7 miles deep. The limiting factor, a submersible built by a private company and used by explorer Victor Vescovo to reach the bottom of all 5 oceans, is one of a handful of vehicles capable of surviving the Challenger deep. These vehicles represent the current frontier. Building a vehicle that survives 6 miles down requires solving several engineering problems simultaneously. The hull must withstand 16,000 pounds per square inch without deforming. A sphere is the most efficient shape for resisting uniform external pressure because it distributes the load evenly in every direction. Both the Fendougé and the limiting factor use spherical titanium pressure hulls for the crew compartment. Titanium is chosen because it has a high strength-to-weight ratio and does not corrode in salt water the way steel does over time. The sphere is machined to extreme tolerances, fractions of a millimeter, because any deviation from a perfect sphere creates stress concentration points where the hull is more likely to fail under load. The viewport, the transparent window that allows observation, is one of the most technically difficult components. Standard glass shatters under deep trench pressure. Viewports for extreme depth vehicles are machined from solid acrylic in a specific truncated cone shape that actually becomes stronger under compression rather than weaker. Every electrical connection, every cable penetration through the hull, every mechanical control that passes through the pressure boundary is a potential failure point. Each one requires a specialized deep-rated seal. Battery capacity limits mission duration. At trench depths, the vehicle cannot communicate with the surface in real time because radio waves do not travel through water. Navigation relies on acoustic systems, which are slower and less precise than GPS. Sample collection tools must work reliably at pressure and temperature conditions that cause standard lubricants to freeze and standard electronics to behave unpredictably. New submersible programs currently in development are focused on 3 improvements over current vehicles: longer bottom time, which requires better battery systems; better imaging, which requires camera systems optimized for the specific light conditions of deep trench surveys; and pressurized sample containers which can maintain deep-sea pressure during ascent so that biological specimens arrive at the surface alive and chemically intact. That last capability is the one that would change the science most dramatically. Currently, every biological specimen brought up from extreme depth experiences a pressure drop from 16,000 pounds per square inch to 14 pounds per square inch. During the ascent, the organisms that survive this transition are either dead or severely compromised. A pressurized sample container would maintain the specimen at close to its natural pressure, all the way to the surface laboratory, allowing scientists to study living deep trench organisms for the first time under conditions close to their natural environment. Confirmed, pressurized sample containers are under development in multiple deep-sea research programs. The organisms at the bottom of the world are ready to be understood more completely. The engineering is the bottleneck, and once that bottleneck breaks, every question that has been stuck at unknown for the last 15 years becomes potentially answerable. The final and most fundamental of those questions is also the oldest. 6 miles down, in total darkness, under pressure that flattens military submarines, a single cell the size of your fist sits on volcanic rock and accumulates uranium without dying. 6 miles down, in a different trench, tube worms with no mouths feed off bacteria that eat poison gas, building an entire layered ecosystem with no sunlight, no surface connection, and no theoretical reason to exist by the rules biology followed for 200 years. These discoveries answer some questions. They raise a bigger one. What is the minimum threshold for life? Life here requires liquid water, an energy source, organic chemistry, and stable enough conditions for complex molecules to form and replicate. That is the working definition, but the deep sea keeps pushing on the edges of stable enough. Xenophyrophores live in conditions that are actively toxic, under crushing pressure, in absolute darkness. And they do not just survive. They grow to record size. They reproduce. They build communities that host other organisms. The chemosynthetic ecosystems run on energy sources and at pressures that 2 generations of biologists would have considered incompatible with complex life. Each time scientists have drawn a line at the edge of where life can exist, life has been found past that line, which raises the question that no current science can fully answer. Is there a line at all? The confirmed record for life at depth is 6.6 miles. The seafloor continues deeper than that. The chemistry that drives chemosynthetic communities exists in principle wherever geological seepage occurs, and geological seepage is not confined to the depths already surveyed. Scientists studying extremophiles, organisms that live in extreme conditions, have found life in volcanic rock miles below the land surface, life in ice, life in boiling acid springs, life in salt concentrations that would crystallize most cells, life in the cooling water of nuclear reactors. Each environment that was once considered impossible to support life has turned out to support life. The xenophyrophore and the trench tubeworm community are not the endpoints of this pattern. They are data points in a trend line that keeps moving. Where does it stop? The honest scientific answer is that nobody knows. The theory that life requires certain specific conditions is consistently tested and consistently found to be more flexible than previous estimates. Each discovery of life in an extreme environment forces a recalibration of where the actual limit lies. Confirmed. Life exists in a broader range of physical and chemical conditions than the pre-deep-sea science assumed. Unknown, whether there is a hard lower limit to the conditions compatible with life, and if so, what those conditions are. The search for that limit is what drives deep-sea exploration, astrobiology, extremophile research, and the missions currently traveling toward Europa and Enceladus. The creature at 6 miles down, the one that should not exist, the one that eats uranium and builds a shell from trench sediment and houses hundreds of nuclei in a single cell, is the reason we now ask the question differently. The question used to be, where might life exist? The deep sea taught us to ask instead, is there anywhere it cannot? And the answer, when it comes, will be the most important thing biology has ever confirmed.